Professor Thomas Neer. He is Professor and Chair of Ecology and Evolutionary Biology, as well as Bingham Oceanographic Curator of Ichthyology, I hope I'm saying that right, at the Peabody Museum of Natural History, and Head of College for Saybrook College. He hails from Chicago, where the long shoreline of Lake Michigan and the rivers of the upper Midwest provided his first explorations of aquatic biodiversity. After obtaining a bachelor's degree in both history and biological sciences, Neer earned his PhD in ecology, ethology, and evolutionary biology from the University of Illinois, Urbana-Champaign. And prior to joining the faculty at Yale, Neer was a postdoctoral fellow at the Center for Population Biology, UC Davis, and an assistant professor in the Department of Ecology and Evolutionary Biology at the University of Tennessee. Professor Neer's research focuses on the mechanisms that generate biodiversity through studying the evolutionary history of fishes. This work focuses on marine fishes in the Southern Ocean surrounding Antarctica and North American freshwater fishes. Professor Neer's research team utilizes genomics and morphology to reconstruct the fish tree of life, discover and describe new species, and investigate mechanisms that result in speciation. At Yale, Professor Neer is the chair of the Department of Ecology and Evolutionary Biology, as I told you, and also curator at the Peabody Museum, and last but not least, head of Saybrook College. So I'm sure he'd like to give a shout out to all of you Saybrook alum. He resides at Biggs House at Saybrook College with his wife Allison and their two daughters, who are ages 11 and 9, which means they must be very into discovering all the kinds of fishes that you know about.
Absolutely.
So welcome, Professor Neer. Thank you so much for this presentation.
Thank you so much, Lauren, for that wonderful introduction. And thank you all for being here today. It really is an honor to be asked to give a perspective on the biodiversity of a group of organisms that dominate a big part of my intellectual life. So it's hard to find work-life balance when you're an ichthyologist, somebody who studies fishes, because I get so much joy, not just from the scientific work, but in all ways that I get to interact with the fishes. And I just want to acknowledge that I think many of you, I saw a lot of the reasons or the aspects of have you interested in fishes in the chat before we started today. And I just want to note that in the years I've been teaching a course called Ichthyology, which covers the biodiversity of fishes, the students who come into the class, we get a lot of non-science majors who take the class, and it's aquarists, people who've kept fish, it's scuba divers, people who've interacted with fishes in marine habitats through scuba diving. Of course, it's anglers, people who like to fish, whether it's fly fishing here, such as in the left-hand corner with this cutthroat trout. Also, people who really got into fish because of phenomenon or programming such as Shark Week, which I think we're in Shark Week right now. And I forgot the cable channel that has a Discovery Channel has Shark Week, which becomes famous. And I'm now seeing students who were so jazzed by Shark Week, they're into fish. So what I want to do today is, first of all, really convey to you just how deep the iconography of fishes are in our culture. Here's a humorous little side that tries to tip my hat both to literature I engaged with as a child, the aspects of fish diversity that motivate my research today, but also the fact that half of all living vertebrates, that is an animal that has a backbone. So we're talking about fish, amphibians, reptiles, birds, and mammals. Of course, humans are mammals. Half of all living vertebrates are fishes. So fishes are really important. And obviously, they're important to the economy and aspects of human health. So what I'm going to talk about today is I want to take this opportunity to introduce you all to the idea of evolution and why evolution is important in that it provides the only set of theory that explains the origin and maintenance of biodiversity. That's it. That is the only theory that's been proposed that allows us to have a scientific explanation of why the planet has the number of species it has, how those species originated. It allows us to speculate about how life originated, but then also explains stuff that we find in rocks, such as fossils, that one of our participants mentioned they were interested in. Then I'm going to tell you about how evolutionary biologists study the history of life. It's through this intellectual and methodological innovation that is only about 60 years old, and that's called phylogeny. That is essentially the concept of a tree of life, but an objective way of inferring that tree of life. And then we're going to get to fish, and I'm going to make the point that we are all fishes, and I'll show you that by understanding phylogeny and understanding the diversity of vertebrates. I will prove to you that we as humans are actually fishes. And then we're going to get into the weeds a bit. I'm going to tell a couple of fish stories. I'm going to talk about this group of fish called the Acanthomorpha. I put a question mark there because only the real fish nerds have ever heard of this group. But when I tell you about them, you're going to be amazed at how important this group of fishes are relative to your familiarity with the term Acanthomorpha. And then I'm really going to tell a fish story, and I talk about fishes in Antarctica, which are unlike any fish fauna on the planet. Now, as Lauren mentioned, I'm from the great city of Chicago, and I grew up in and around Lathrop Homes, which is a housing project, was a housing project on the north side of Chicago. In fact, it was the furthest north housing project in Chicago. It was built in the 1930s under Roosevelt's WPA program. It is unlike, some of you may be familiar with the consequences of the devastating urban renewal that happened in Chicago after World War II through the 50s and 60s, where there were large tracts of housing projects that were built and surrounded by interstate highways to keep people away from and contained to certain parts of the city. While this was not the easiest place to grow up, it certainly, I had resources as a child growing up in this environment. I had actually quite outstanding public schools. I went to Yon Elementary School, and then also public institutions such as the Shedd Aquarium, the Field Museum of Natural History, and the exhibit-based Museum of Science and Industry, really to fuel my curiosity of science when I was a child in elementary and high school. But also, as Lauren mentioned, I was really into aquatic biodiversity. And I'll point out Lathrop Homes here is on the bank of the North Branch of the Chicago River. And I could tell you, when I was a kid, I believe the year was 1980, I landed a smallmouth bass here in the Chicago River, right there in the projects. And there was no better way to get street cred in Lathrop Homes than catching a big smallmouth bass in the Chicago River. And I saw that for me, it was a great illustration of how fish brought people together. So growing up in Chicago at times was rough, but I had great public institutions helping me out. I was a winner, as well as a participant and a winner of the city, Chicago City Science Fair, All City Science Fair when I was in 8th grade. And after high school, I was recruited at Northern Illinois University through a program called Chance that targets students from disadvantaged backgrounds, primarily from Chicago and Rockford, Illinois. And after that, I was well on my way. I finished my PhD work at the University of Illinois, where I was working on the evolutionary biology of North American fishes. Then I had this outstanding opportunity to obtain a very competitive postdoctoral fellowship at UC Davis, and then landed a job at the University of Tennessee. And then after a couple of years, I was recruited by Yale. That was 15 years ago. And Yale has been a remarkable place to live and develop my career. At Yale, I wear many hats, as Lauren mentioned, and these are all, in a sense, intersecting and almost equally important. I'm, of course, a faculty member and chair Department of Ecology and Evolutionary Biology. This is where I do my research and my teaching. I curate a ever-growing research collection of fishes at the Peabody Museum of Natural History. And then Yale's grand surprise, or I should say the grand detour in my career, was being asked by President Salovey to come in as the head of college at Sabre College, a college of which I was a faculty fellow. And I didn't come to Yale to get into student life, but this experience, 6 years now, has been transformative. And it has given a space where essentially my wife and I, Allison Neer, who's an attorney here in New Haven, where we raised our 2 children. They were 3 and 5 when we moved in. And being the head of college at Sabre has been a remarkable experience. And I'll just brag a bit that, yes, we were 3-time three-peat champions of the intramural Tin Cup, and we're going for our 4th this year. So the core of what I do is evolution. Now, evolution is a hot button topic in some parts of the world, maybe even among some of you here in this audience. So here is Gary Trudeau, Yale alum, has this great cartoon. I'm going to paraphrase. So basically, the doctor says, uh-oh, I hope he's only a Sunday creationist after looking at his x-ray. And he tells him, you know, you have tuberculosis. So the outlook's good because we're catching it early, but he asks, are you a creationist? He says, why do you ask? He says, because I need to know whether you want me to treat the tuberculosis bug as it was before antibiotics or as the multiple drug-resistant strain it has since evolved into. Patient asks, evolved? Doctor says, your choice. If you go with the Noah's Ark version, I'll just give you streptomycin. What are the newer drugs like? The patient asked. Doctor answers, they are intelligently designed. And the point of sharing this with you is that evolution, not only, as I said, provides the only basis for understanding, scientific understanding of the origin and maintenance of biodiversity, but is a central core of medicine as well. Okay, so evolution has a really interesting history. Now, we know, okay, Copernicus put the chink in the geocentric universe, right? Think about a heliocentric universe. And Galileo was a copious author, many of which has survived the time that he lived. There are no photographs of Galileo or Copernicus. There are photographs of the people who came up with evolution. In fact, when the first book, Darwin's book, was published, I had a great-great-grandparent that was alive. That's not that long ago. So our understanding of the development of this idea is really sophisticated because it happened fairly recently. And I would argue that evolutionary biology, evolutionary theory, it was one of the last great abstractions of humanity with regard to the natural world. People had an understanding, in a sense, almost of how the universe is constructed before there was a scientific explanation of biological diversity. So let's take a historical setting. So let's look at the end of the 18th century. So we're coming up upon 1800. Most people in Europe and the Western world thought the Earth is only thousands of years old. Species cannot change. They're immutable. They are made by a benevolent creator and they never change. And all species that ever existed are currently living somewhere on the planet. There's been no extinction and no generation of new species. And adaptations, that is when we see an organism that seems to have a fit to its environment. Say a pollinating moth that has a long proboscis to reach into a deep pollen tube of a flower, or nectar tube of a flower, we would argue we think of that as an adaptation. Prior to evolution, Europe and the Western world thought that those were produced by divine intervention. Now, Darwin publishes his book, and I'll tell you a little bit about Darwin. So he published his book in 1859, but Darwin was not alone. And again, we know this because 1859 is not that long ago. This is very well documented. So there is, in a sense, a new scientific revolution happening in the 19th century. So Charles Lyle, a geologist whose book, The Principles of Geology, has never gone out of print since its publication between 1830 and 1833. There were 2 volumes. Lyle says the Earth is very, very old. And he utilizes geological formations and the fossil record to make this argument. Fossils represent extinct species. So this becomes part of the scientific conversation. And people are even starting to argue, particularly French scientists, that existing species are descended from ancestors whose relationships we could figure that out based on traits that they have in common. So the way Cuvier, who by the way was an ichthyologist, would argue that all vertebrate animals share a common ancestor is by the fact they have a backbone, they have vertebrae. And all vertebrates also have a cranium. They have traits that they share in common, and those traits shared in common are indicative of their common ancestry. And then Darwin finally comes in and essentially distills and summarizes all of this conversation, as well as his own scientific observations, and adds a mechanism to explain adaptations, which are famously known as natural selection. I study fish. So if there was no evolutionary biology, I would have no explanation to explain why all of these really divergent species of fish, all of which you could find in the state of Tennessee, all of which are closely related to each other, I would have no scientific theory to explain why are there so many different species of darters, and why do these different species of darters look the way they do, and why do they have the biology they do? Also, if there was no evolutionary biology, I would have no scientific explanation to explain why there are so many species of freshwater fishes in North America relative to other non-tropical regions of the planet. So here's the list here. So North America basically crushes all other temperate non-tropical regions in the world with regard to its species diversity. So its evolution is not aimed at the argument that humans are primates. We are primates, but evolution goes well beyond that in providing a set of theory for us to develop explanations of why North America has such rich biodiversity when it comes to freshwater fishes relative to other parts of the planet. And also provides an explanation why in that Doonesbury cartoon, the physician should not be using streptomycin to treat tuberculosis for his patient. Let's talk a little bit about Charles Darwin. So I have his birth year here. I was my fancy attempt at animation. His head will pop up in a moment, but I just want to point out that I'm really interested in human history. And many of you may not know that Charles Darwin and Abraham Lincoln were born on the exact same day. They were both born on February 12, 1809, which I just find a really amazing coincidence that one of the most transformative figures of the political world of the 19th century was born on the same day as one of the most transformative figures of the scientific world of that century. So Darwin he wants to figure this out. He wants a scientific explanation for why there are the species there are, calls it the species problem. So while he's circumnavigating the globe on his famous voyage of the Beagle, he reads Lyell's book that says the Earth is really old. And Lyle essentially lays out the fossil record for Darwin. So Darwin is not just a deep thinker and pulling ideas and observations from others. He makes his own observations. He dug up extinct armadillos, giant armadillos in South America, fossils that were in the same geographic region as their living relatives. So not all species that existed are on the planet today. He finds marine fossils uplifted several thousand meters in the Andes. If this is a high alpine Andes, what are marine fossils doing way up here? The Earth has changed. The Earth is dynamic. He sees the effects of a massive earthquake in Valparaiso, Chile. And he gets an idea that dramatic changes to the landscape can happen rapidly in a short period of time. And then, of course, his famous visit to the Galapagos Islands, he finds different species of closely related lineages on different but adjacent islands in the Galapagos. So why would a benevolent creator put a different species of mockingbird on each of the different islands of the Galapagos? So Darwin starts thinking that maybe populations become geographically isolated from each other and then eventually diverge to become different species. While he didn't have the mechanism, the theory is spot on with 21st century understanding of how species originate. Then he returns to London. Darwin was a person of means, so his job was basically to just do science. He wasn't employed by anybody. He reads an essay about the growth of human population by Malthus, and he puts that together and he comes up with a mechanism of natural selection. There he is. He pops up on the screen now. So in 1859, he publishes his book on the origin of species. The publisher ran 1,250 copies. It was sold out the first day. That is, the booksellers, the middle people of the book business, basically bought all the available copies of the publisher. Eventually, 6 editions were published. If you ever want to take a dive into Darwin, he's actually a really good writer. I strongly encourage you to go with the first edition, 1859. It's still in print, is carried by Harvard University Press. And also it's available online. It's public access at this point. So what's amazing about evolution in Darwin's formulation of it is that 1859, think about modes of communication. Okay, news came from Europe and news from North America made it to Europe, and news got around the world, but it happened a lot slower. Almost overnight, essentially, Darwin's idea is universally accepted by a biologist. Biologists everywhere read Darwin and said, oh yeah, that makes perfect sense. I now have a centralizing set of theory to explain why there's so many bees or why bees and flowers interact the way they do, et cetera, et cetera, et cetera. There's a couple of notable holdouts, one of which is a Harvard professor by the name of Louis Agassiz. So I always drop that joke when I teach here at Yale. So the one famous holdout globally to Darwin's work was a Harvard professor. Okay, Darwin has 2 theses, and the first one is really important for us. And I'll mention the second one briefly, but we're not going to go into that. This is so revolutionary, and it's never been refuted. No one has ever been able to reject this hypothesis, and that all life on Earth shares a common genealogical origin. All life on Earth is intertwined in a tree of life. This is a radical idea at the time, and to some people today is still a radical idea. He refers to this as descent with modification. You may have heard that phrase when Darwin is referred to. And this is, he's refuting kind of special creation of species or creation in general. And then if we got into the weeds, a mechanism of evolution that was proposed by a fellow named Lamarck. Now, I just want to take a moment, the only thing I'm going to read to you today, just to illustrate Darwin's gift as a writer. Now, he's a scientist, but he's trying to pull you in as the reader to link evolution as a mechanism that is akin to physical processes that are keeping the moon in orbit around Earth, with some Victorian flowery prose. There is grandeur in this view of life, with its several powers having been originally breathed into a few forms or into one, and that whilst this planet has gone cycling on according to the fixed law of gravity, from so simply beginning, endless forms most beautiful and most wonderful have been and are being evolved. So there it is. He is rhetorically linking mechanisms of evolution to the mechanisms of the Newtonian universe. Brilliant and well done, in my opinion. Now, the second of these 2 theses are that natural selection is the main driving force for adaptation. Now, natural selection is certainly important, but natural selection is not the sole driver. And this is the thesis of Darwin that has been refuted. We know evolution can happen without natural selection. One mechanism I'll just mention, we won't be able to go into details, called genetic drift. Genetic drift is where gene frequencies and populations change just given the chance sampling of the parental genotypes that come together to form the offspring. Okay, now going back to Darwin's first thesis, this is illustrated in the only figure in the 1859 volume. Now, in the 21st century, we refer to a branching diagram that relates the relationships of organisms as a phylogeny. So what Darwin is showing in this figure are ancestor-descendant relationships where the top line is the present day. And those Roman numerals, as you go down to the bottom of the figure, Roman numerals decrease. Those are deeper and deeper periods of geologic time. And he's showing that some lineages go extinct, others persist. You have diversification, and everything at the top here, Roman numeral 14, that's alive, essentially has ancestry relative to the lineages in the past, as well as relative to each other. This is really important. What Darwin is saying is that, you know what? If these organisms share a common ancestry, we might be able to figure it out. In the same way a chemist would bring in, a complicated mineral and be able to tell you what atoms are involved with those crystals, there's a method to that. What Darwin is saying, it's going to happen, but I'm going to be long dead when we figure it out, that there will be an objective, data-driven way where we can infer that tree of life. Now, the way the tree of life was inferred in Darwin's day, from Darwin's day till about the 1960s, was the expert would say how these lineages are related. And what I, as an ichthyologist, thought were important would be different what, say, an ichthyologist at Princeton thought was important, which would be different than an ichthyologist maybe in Japan would think was important. So different scientists could be working on the same group and come up with different phylogenies. That's not really the case today. And I'll explain why. Because what we're doing, everyone who studies phylogeny, we're not all fans of Elvis, that's not what I'm saying, but we all use a same set of data-driven objective methods to infer the tree of life. Okay, so in this example, we have Elvis representing our species, we have a mushroom, which is a fungus, and then we have it's a really interesting lineage of flowering plants. It's an angiosperm. So we want to know, of these three lineages, if they share common ancestry, that's Darwin's first thesis, which of the two are more closely related? Now, the reason why different scientists came up with different phylogenies is they emphasize different things. So we could say, oh, we're going to take Elvis, because at least in his Viva Las Vegas era, was svelte and golden. And we would say that the angiosperm, the flowering plant, is skinny and golden as well. So we think those two are more similar. So we're going to kind of group those together. Or we might say, well, both the flowering plant and the mushroom are sessile. So maybe we want to group those together. And Elvis, that's why he was controversial, at least before his Las Vegas days. He was anything but sessile, right? So he was always shown from, like I am today, from the chest, shoulders up. So we may say that's a phylogeny we like. But actually, the philosophy behind phylogeny, and this is why there's an objective universal approach, is we use the principle of thinking of relationships that are based on the shared evolutionary history. So indeed, I was just at the cusp of mushrooms being thought to be plants, where fungi were always taught in botany classes. But indeed, fungi and animals are more closely related to each other than either is to plants. So this would be the phylogeny. Let's use another example with Elvis as our human. And this one actually blew up in a heated controversy from an exhibit at the British Museum, now called the Natural History Museum, in 1981. So we have a lungfish. There are three lineages of lungfish. We're looking at the Australian lungfish here. We have a salmon, and we have a human. And we could say, well, the Australian lungfish and the salmon obviously are both fish. They have gills, they live in aquatic habitats. You put them on land for a while, even though the lungfish can gulp air, they're going to die. They need to live in water. So we would say that probably the lungfish and the salmon, they should be grouped together because they have a similarity based on their fishiness and their ecology. Well, if we actually look at the evolutionary relationships, the human and the lungfish, that is any vertebrate that has four limbs, so that includes amphibians, reptiles, birds, and mammals, turtles, share common ancestry with the lungfish relative to the salmon. And this is why we are all fishes. We are all fishes because if we look at a phylogeny of all the lineages of vertebrates of which we are aware, everything in green is a fish. Everything that's in a gray box surrounded by a green line is an extinct lineage of fishes. Anything that's yellow is a tetrapod vertebrate. So any frog, salamander, snake, lizard, turtle, bird, mammal, primate, canine, carnivore, rodent would all fit in that yellow box on this phylogeny. So we indeed are more closely related to fishes, some fishes, than they are to other fishes. So if you close your eyes, unless if you're a shark nerd, and I ask you to envision a fish, you're going to think of a fish that's a rayfin fish down here. So us and rayfin fishes are more closely related to each other than either is to sharks, skates, or rays. So we're all fishes. And when students at Yale take ichthyology, I say, you made the best decision of your biological career because, to date, because you could take Professor Prum's ornithology class, which I think is the best science class taught at Yale. But by taking that class, you're just getting a little tiny twig of this yellow box. By taking ichthyology, you're getting the entire diversity of vertebrates. So we are fishes. And that point is emphasized by this wonderful book by my colleague, Neil Shubin at the University of Chicago. I'll give a shout out at the end of the talk, called Your Inner Fish. Now, what I do is I use phylogenetics to study fish. So I'm interested in the mechanisms that are producing new species in North America, and I actually describe new species. I'm involved with constructing the big fish tree of life. And I'm also interested in how evolution has worked in the context of climate change with fishes in the Southern Ocean surrounding Antarctica. Okay, so on to our deeper dive with fishes. So acanthomorphs, wow. Acanthomorphs, as you can see here, include a full range of fishes that many of you are familiar with. So, but they're probably the most species-rich group of vertebrates you've never heard of. So there are about 63,000 species of all living vertebrates. 19,500 are acanthomorphs. Nearly 31% of all living vertebrates are this group of acanthomorphs. Acanthomorphs are the group of fish that completely dominate marine habitats. And basically, we have had incredible difficulty resolving the tree of life of these fishes using traditional morphological characters. In fact, this is what the tree of life of acanthomorphs have looked like for the past 50 years or so. Really uninformative. You have multiple lineages coming out from the same part of the tree. You don't have a branching nature here. So what we've done is we've been sequencing genome scale data sets. And with that massive amount of data, we're able to finally figure out the architecture of this phylogeny. And what's really interesting about this, there have been some remarkably radical resolutions, changes to how we think these species are related that have had dramatic impact on how we envision their biodiversity. So first of all, I know everyone here is familiar with anglerfishes. Anglerfishes are a trip. They are so weird, mostly deep sea, particularly the deep sea piscivorous anglerfishes. And this little thing hanging off this fish is a male. This male is a parasite. So the male bites onto the female, his face and brain basically grow into the female's body, their circulatory system become one, and he essentially is a gonad. He is basically only there to produce the male gamete, the sperm. So last summer, the code was cracked. How can this happen? Well, it turns out anglerfishes have lost a number of genes that are involved with adaptive immunity. So if a person has an appendage transplant or an organ transplant, the adaptive immunity is working against that person, right? That adaptive immunity that keeps us, protects us from pathogens will also work to rejection of those tissues. So anglerfishes have figured out how essentially to do organ transplantation. They've essentially turned off parts of their immune system. And I know everyone here knows pufferfishes, right? They're super cute. And the ocean fish, mola mola, puffers and ocean sunfishes are closely related. Well, it turns out anglerfishes and pufferfishes share common ancestry relative to all other vertebrates. They are two closely related lineages in the fish tree of life. This was completely unanticipated and verified through several independent analyses of different aspects of the genome. Another unexpected relationship, particularly if there are some open ocean anglers here, you may have been targeting tunas and billfishes, such as marlin and swordfish. For about 250 years, everyone has thought that tunas and billfishes are closely related. They're not. They resolve in really different parts of the fish tree of life. In fact, billfishes are related to barracudas as well as flatfishes. So the marlin is closely related to the flatfish relative to the tuna. And this has given us insight into the evolution of flatfishedness. How do flatfishes do this? How do they become asymmetric, where one eye basically migrates to the other side of the head during their embryonic development? Tunas, super important food fish, right? Turns out tunas are closely related to seahorses. Nobody really knew. Well, people thought seahorses were related to this group of fish called sticklebacks, which you could catch in nearshore habitats here in New England. Tuna and seahorses actually share a very close relationship. So think about the crunchy seahorse next time you're biting into a piece of sashimi when you're at a Japanese restaurant. So the architecture of the tree of life has emerged and has allowed us, which has emerged primarily from the analysis of genomic data. Morphological data did not really give us a tree of life for fishes the way it had for other groups of vertebrates, such as amphibians, reptiles, mammals, and birds. Now, of course, genomics have been applied to the phylogenies of all those other groups, but for fishes, it radically transformed our understanding of their relationships. Okay, so now it's time to tell a fish story. And I mean, fish stories are great, aren't they? I've told several in my life. And the story I'm going to talk about is how climate change fueled really remarkable species diversity we find in Antarctica around the Southern Ocean. But I'm going to take a cue from someone who at least became a resident of Connecticut. Granted, he's from Missouri. He says, don't tell fish stories where the people know you, but particularly don't tell them where they know the fish. So I don't know who's on this call who knows me, but I'm joking, hoping that some of you or you are not familiar with Antarctic fishes, so I could tell my fish story. The story of Antarctic fishes is the story of the exploration of Antarctica, which was fueled by, peak imperialism, essentially. Western powers were looking for a southern route, basically to get to other parts of the world. And Antarctica was a prime target for the exploration of natural resources. So there was a spurt of activity from the 1880s through the end of World War I. Then it picks up again, but with definitely more of an international collaborative flavor in the 1950s. So this is Ernest Shackleton, Sir Ernest Shackleton, intrepid British explorer, whose ad here for one of his famous journeys says, "Men wanted," of course, it would say people wanted today. Indeed, some of our heroes of Antarctic discovery over the past 50 years have been women. But I love this ad. It says, "For hazardous journey, small wages, bitter cold, long months of complete darkness, constant danger, safe return doubtful, honor and recognition in case of success." It reminds me of my days in graduate school. So they set out on their ship. The ship becomes caught in the sea ice of the Weddell Sea, and the ship is crushed. So they have to traverse a couple hundred kilometers over the sea ice to an archipelago on the western side of the Antarctic Peninsula, so due south of South America. And Ernest Shackleton and the skipper of the ship get into an open boat, a whale boat, basically, a whaler, and sail to South Georgia, which is on the east side of the peninsula at the edge of the Weddell Sea. And they get there and they land on the wrong side of the island. They have to traverse a mountain range that people had never traversed before. They come down. It's a whaling station there. They get a ship to go rescue the compatriots that were left on Elephant Island. They were there for a year, 1 year, on a literally deserted Antarctic island. And nobody died, not a single fatality. There were several lost toes, but no fatalities. It's a great story. Now, right away, it became clear that there's a really interesting biodiversity there. And there's this group of fish. There's no common name. They're called Nodothenioids. It's about 120 species. And if you're familiar with fishes, they're actually related to these darters I work on, sea basses, sculpins as well. And there's a lineage of about 100 of these that are around Antarctica that we call Nodothenioids. And there's about 100 species of those. And they have these remarkable potential adaptations to polar conditions. Now, some of you may be familiar with Nodothenioids if you've ever eaten Chilean sea bass. By the way, the official common name for Chilean sea bass is the Patagonian toothfish. You can see the teeth here. But a New York chef said, I can't market toothfish, and came up with Chilean sea bass. There's some issues about overharvesting of this long-lived, late sexually maturing species. But if you've ever had Chilean sea bass, this is not a guilt trip. It is a way to say you have encountered an Antarctic Nodothenioid. Now, Antarctica is a really interesting part of the world in that it's really different from the Arctic. You could think of Antarctica, it's a continent surrounded by an ocean. It's called the Southern Ocean. I think National Geographic now puts the Southern Ocean on their stamped globes. The Arctic, you could think of as a small inland sea surrounded by continental land masses. So oceanography is completely different. And Antarctica has really the only circumpolar current, the only circular current on the planet, known as the west wind drift. So as Antarctica became cold, it kept getting colder because it was basically immune to warm water currents that would have come to the continent. Now, the story of Antarctica is one of climate change. Antarctica was not always the frigid polar place it is today. It's a story also of the fragmentation of Gondwana, but the eastern part of Gondwana. So we're looking at here is the reconstruction of temperature through time. So this is in millions of years. So right at about 35 million years ago, the world, and Antarctica in particular, went from a warmer climate to a much colder climate. And what we see in Antarctica is that about 15 million years ago, you really start getting the cold, freezing polar conditions that characterize that part of the world today. Now, the fishes of Antarctica are wholly bizarre in that one aspect is that if we all could take the Yale alumni dream trip, I would take you all over the planet and we would catch fish in nearshore marine habitats. And if we did that, we would catch representatives all along that fish tree of life I was showing you a few slides ago. Because we're good ichthyologists. We're going to catch these fishes that represent all these major lineages. In Antarctica, it's the only place on the planet where the entire marine fauna of fishes on a continental scale is dominated by one tiny group of closely related species, okay? So that is unlike anywhere else. They represent about 76% of the species diversity, this group, the Notothineoids. They represent about 92% of the abundance, meaning the number of individuals you encounter. And they represent 91% of the biomass, meaning if we weighed every fish we caught, completely dominated by these Notothenioids. But this was not always the case. Antarctica in the past, when it was a warmer part of the world, it had a fish fauna that's very similar to basically Long Island Sound. And this is from a fossil formation at the tip of the Antarctic Peninsula called Seymour Island. And in fact, you have fishes that you tend to find in tropical coral reefs, such as wrasses, the Labridae. But if we look at the fishes that are around today, they're basically the only thing that's still with us in the Southern Ocean are these skates and rays and the Nodothenioids. How are they there? And what happened to all those other fishes? Well, the onset of polar conditions, it just got too cold. These fishes literally froze. And with these Notothineoids, we have a really good working phylogenetic tree. And the Antarctic Nodothenioids are all closely related to each other relative to other Notothenioids. And then we have this fish, Elaginops maclevinus, which is found in southern South America, never been exposed to polar conditions. And it is the living sister species to this entire lineage of 100 Antarctic species. And it's not as if we missed other species here. Elaginops maclevinus, that's it. It's this weird singleton lineage that is the sister lineage to these Antarctic fishes. So yellow for the non-Antarctic, blue for the Antarctic in the few slides leading to the end of this session. Now, this is why Notothenioids are the only fish in the Southern Ocean. This fish is resting on the interstitiary bottom of the sea ice that forms, the surface ice. So this water is negative 1.86 degrees centigrade. If you touch that ice with your bare hand, your tissues would freeze solid because that super cold water is basically going to transfer ice crystals to your tissues and you would freeze. This fish lives on, this is its surface. Why is it not freezing solid? It has the same number of solutes in its blood that we do. Well, what's going on? So this is how the fish should look. It should be encased in ice. And that's what happened to all those other fish lineages that were in Antarctica. They essentially got froze out, literally. So if you take this fish that would freeze solid and you look at its body fluids, yes, there is salt and other small ions that depress the freezing point. So this is the freezing point here. Water freezes at 0 degrees centigrade. With salt and small ions, with your normal fish that lives in the temperate part of the world, yes, that body fluid would freeze at negative 0.7. But remember, we have to go to negative 1.86. And if we look at an Antarctic Nodothenioid, we see that the salts, as well as this protein called antifreeze glycoprotein, an antifreeze protein, gets their freezing point all the way to negative 2.2 degrees centigrade. Now, for any chemistry nerds in the room today, the protein is a very simple alanine, alanine, threonine amino acid repeat, and that triplet's repeated multiple times. And at the threonine, it has a 2-sugar side chain, a disaccharide side chain. And the way this protein works is you're looking at the protein or the amino acid backbone and the sugar side chains. The sugar side chains basically interact with the lattice of the growing ice crystal, and they stop the ice crystal from growing. These fishes avoid freezing. Now, these fishes also do something that no other vertebrate does. No other vertebrate. There's no exception to this. This is a subgroup of these Notothenioids called ice fishes. They are scaleless. They have no scales, and their skeleton is mostly cartilaginous. This fish here, you could see, it's almost a half a meter long. And what it looks like is if you took an embryonic fish and blew it up to adult size. These fish look like embryos. They look like adult size embryos. Now, what makes these ice fishes unique, there's only 16 species of those 100 species of Notothenioids, is that they are the only vertebrate animal ever known to lack hemoglobin in their blood. They have no oxygen transporting molecule in their blood. So here is the blood of an ice fish. It's white. They have no red blood cells. Here is the blood from a non-ice fish Notothenioid. It's red. It looks like our blood. It has red blood cells that carry oxygen for respiration. They do all of their oxygen transport through passive diffusion in their blood. And they do it through their scaleless body. They're able to get diffusion, highly vascularized capillaries at the body surface here. Now, this is the only place on the planet this could evolve. And how it evolved is, without going into the details, they've essentially lost the genes associated with making hemoglobin. And the water is so cold in the Southern Ocean, it's saturated with oxygen. So it's the only place in the world that this could happen. Now, if we look at the Antarctic Nodothenioids, they're not 100 species of ecologically the same thing, they're really different ecologically. And here's a cross-section of a nearshore habitat at McMurdo Sound, where the big US research station is. We have large pelagic predators. This fish here can get up to 1.75 meters. That's a big fish. And this fish eats other pelagic fishes. You have fishes that are on nearshore benthic habitats. And all Notothineoids lack a swim bladder. The swim bladder is a structure that has an ancestral origin from lungs. So a swim bladder in a fish is the same as a lung in a tetrapod vertebrate. And the swim bladder is the organ a fish uses to inflate and deflate to regulate its buoyancy in the water column. These Notothineoids do not have a swim bladder, but yet they've evolved these big pelagic predators that are utilizing water column habitats. Well, it turns out these pelagic fishes have the unique lipid deposits in their body. They put fat in to increase their buoyancy. But Antarctic Nodothenioids, what we're looking at down here in the bottom right-hand corner, these are CT scans of bone density. So hot colors are dense bone, cool colors are very diffuse bone, non-ossified bone. The Antarctic species have lost ossification of their bone. So it seems losing your bone density allows you to be in the water column, may be an adaptation that explains why they're so diverse ecologically. And in evolution, we could call this a key innovation. A key innovation is a trait that evolves that allows a lineage to utilize a set of resources and habitats that other lineages cannot. So a famous example are these columbine flowers that are along the Pacific coast of North America, going from California up into Canada, British Columbia. They have these really elaborate nectar spurs on their flowers. And sure enough, they are designed for very specialized pollinators. And they've made a large number of species and they've speciated in a short period of time. So this is considered a key innovation, allowing a lineage to do something other lineages cannot. Now, this gets into semantics, but the evolutionary history of when this happens is important. If it's an adaptation, it's a trait that natural selection is shaping. Natural selection saying, hey, Notothenioids, reduce your bone density, you can utilize water column habitats that no other fishes are utilizing. Or it could be what's called an exaptation, meaning the trait was already there and it now be able to utilize once the environmental and ecological conditions changed. Probably the most famous exaptation are feathers in birds. So we would think, if you ask a person on the street, why do you think feathers evolved? They would say, oh, well, birds fly. It may be thermal regulation, but definitely fly. Well, feathers are incredibly important for flight birds. However, here is a phylogeny, an evolutionary tree of living birds and all of their dinosaur ancestors. And if we look at the presence of feathers in non-bird dinosaurs, feathers evolve before the evolution of flight. So feathers didn't necessarily evolve for flight. Feathers were co-opted for flight. Feathers were already existent. Now you can see it's really important to make that distinction. So that's what we tried to do here. And what we found just by looking at the evolutionary tree, as well as the bone density, we see that, remember the non-polar South American species? It has really unossified bone. So this idea that polar conditions drove this trait does not seem to be supported by the fact that the non-polar species has the trait as well. And when we dug in, we found that genes in the genome, many of these which are associated with human osteological diseases, were changing at a high rate in these fishes. And what we did is we said, let's look at the genome of all these fishes. And here is their phylogenetic tree. And this colored chart is just a presence or absence of the genes. We found that many of these genes are missing in these species, including the non-polar species. So these genes were lost before Antarctica got cold. And we said, can we replicate the phenotype? Can we make a fish that is not a Nodothenioid have a skeleton that looks like a notothenioid by manipulating the genome of this fish to match the notothenioid genome. We did that using a technology called CRISPR-Cas9. And we did it with kind of a lab rat of fishes called the zebrafish. So we make a zebrafish that has the notothenioid genotype. And sure enough, we get zebrafish that have skeletons that look like the notothenioids. So we've been able to really isolate the genomic aspect that makes notothenioids the way they are. What we also found is that this group has a high rate of genomic evolution, but that high rate of genomic evolution, again, involves the non-polar South American species before Antarctica got cold. And we could put this into a whole context and we could ask, is this amazing ecology we see among these Antarctic fishes, given they're the only fish group in those ecosystems, was that the result of adaptation, natural selection driving it? Or was it an accident, contingency, meaning they had a set of genotypes and phenotypes, the environment changed due to climate change, and they're like, oh, awesome. We've got all these open niches that we can expand in because we came equipped to expand. The open niches are not changing our genotype and phenotype. Those changed prior to the arrival of those changes. And that's exactly what we conclude. So exaptation, as I described it, or thinking a contingency, like evolution responding to accidents or just changes, unanticipated changes leading to what we would characterize as evolutionary success. And actually, when we think about all these traits that make these Antarctic fishes really interesting, they evolved 20 million years before the onset of polar conditions in the Southern Ocean. So they were likely around for a long time. So I want to thank everyone for your attention today and for my foray into a very, very small part of fish diversity. Believe me, I could have talked the whole hour just going group by group through this wonderful fish tree of life. There's so many tales to tell, so many fish stories with the biodiversity of fishes. But what I want to do is I want to leave you with some suggested readings. These are all really incredible works. So I mentioned Your Inner Fish by Neil Shubin, who really makes that case that we are fishes. There's a really wonderful story of the discovery of a coelacanth. It's a lineage that was thought to have gone extinct 80 million years ago, 1930s. They found a live one in South Africa. This is a really wonderful book, A Fish Caught in Time by Samantha Weinberg. If you're interested in how conservation, biological conservation, and politics intersect, I can't recommend more The Snail Darter in the Dam, about the infamous snail darter case in the Endangered Species Act in the United States in the 1970s. And then a book that was out and caused quite a buzz last summer called Why Fish Don't Exist. It's based on that premise that we are fish. But this is a really interesting memoir by a Lulu Miller, who explores the life and really uncovers, it really learns a lot about a very famous ichthyologist named David Starr Jordan, who was the first president of Stanford University. All of these books, or any one of these, would be a great foray to expand your interest in fishes and hopefully pick up on some of the threads that we touched upon today. So thank you again for your attention. I'm happy to take any questions.
Okay, thank you, Professor Neer. Wow, that was such an incredible, it was like traveling through time.
Thank you. And
Back to the origins of
I couldn't even bring
species.
fossils into it, but next time.
Well, we have an opportunity to talk about fossils and anything else that you might want to talk about
Absolutely.
in the Q&A portion of our presentation, which begins now. So if you have questions, please do place them in the Q&A box at the bottom of your screen. There were a couple of questions that have come in already. Erica wanted to know if you would discuss the influence of Alexander von Humboldt on Charles Darwin.
Yeah, so Humboldt is really important in characterizing different floras and faunas. So he was an intrepid explorer, and did a lot of work in South America, and also a fellow named Wallace, who kind of shared credit with the discovery of natural history, was doing similar work that we call biogeography. And so Humboldt did a couple of things. One, showing how plant communities change as you go up the Andes, but also pointing out, the plants you find in the Orinoco Basin are different than the Amazonian Basin. And so Darwin engages with that work, saying, yeah, like what I saw in the Galapagos, but on a much smaller scale, like why is one mockingbird on one island, another one on another? And so a big part of the discovery and articulation of evolution was demonstrating that biodiversity is not uniformly distributed on the planet. And Humboldt was a huge innovator in that area called biogeography. Wallace was as well, and Wallace was focusing mostly on the paleotropics, the transition of plants and animals as you go from Southeastern Asia to Australia. And it's actually a demarcation called Wallace Line that cuts right through Sundaland, like where Indonesia is, where south of that line, it's an Australian fauna, north of that line, it's an Asian fauna. But both in their biogeographic work was really important lines of evidence for Darwin.
Okay, we have a number of questions coming in, so I'm just going to move through them. This is on the Darwin topic. Gary wants to know, what was the biological precursor to fish? Do fish and other sea creatures have a common ancestor?
Yes, so all animals have a common ancestor. So animals, the scientific name for animals is Metazoa, and meaning animal-like cells that are multiple. And that's a thing that characterizes animals, is that we are not colonial. Now, there's a really interesting, if you look at, if we were to sample the genome of every animal alive and make a phylogenetic tree, they would share a common ancestry. So that would be sponges, comb jellies, jellyfish, mollusks, arthropods, vertebrates, starfishes. They would all share common ancestry. The lineage they're most closely related to, they're called Coanoflagellates, and they are colonial. In their cells, they have a very particular type of cell called a choanocyte, and it's the same type of cell that you find in sponges. But sponges are not colonial. Like colonial meaning each cell has its own fate, and it's like, hey, we're hanging out, we're doing our thing together. In non-colonial, like metazoans, the cells are not colonial. They have a tied shared fate and a single developmental origin. So if it's an animal, they share common ancestors. Now, the precursor to fish, that's an excellent question. So we're actually rather fortunate that two bizarro lineages have survived. One is called hagfish and the other is called lamprey. Both are fishes, and they are vertebrates that lack jaws. And they don't lack jaws because they lost jaws. They lack oral jaws because they've never had them. And it's thought hagfishes may be the living sister group to all living vertebrates, including all those extinct things I showed in the gray box. So what you could do, and this is a great question, you could take everything that vertebrates, fishes have in common, and you could trace it back in the phylogenetic tree to that inferred point of common ancestry, and you could try to put together what were the parts that were likely shared by this organism that is the hypothetical common ancestor. And it probably looks something like a hagfish. A jawless vertebrate was the precursor to fish.
So if we follow on that question, would it be safe to theorize, and or do we know that life originated in water or versus on land?
Yes, life originated in water. And here's something that is really cool and blows the mind of first year and sophomore students who take the intro biosequence that we teach here in ecology and evolutionary biology, is that when you look at the terrestrialization of life, when life moves onto the land, So you think about like, what are all the big groups living on land? Plants, obviously, arthropods, insects, and vertebrates, right? All three of those groups come onto land at about the same time in the past, a time called the Devonian, about 300 to 350 million years ago or so.
Interesting. So
Yeah,
plants, insects, and vertebrate aquatic life.
they all started terrestrializing, moving to land around the same
time.
time. And that's because there's this wonderful, like life's most important invention, other than life, is photosynthesis.
Mm.
All photosynthesis comes from blue-green algae. So blue-green algae, they pop up about 3 billion years ago, and the atmosphere essentially had no oxygen when life first originated. So blue-green algae come up, and what does photosynthesis produce? A byproduct, oxygen. And so blue-green algae are the result of the start of the oxygenation of our atmosphere, which then it takes another 2 billion years for that time where the terrestrialization starts happening.
But it makes terrestrial life possible.
Exactly.
Okay. So that answers Ken's question, which was, what about plants? So I want to move to, I think that topic of blue-green algae and how it created oxygen in the atmosphere is really salient to the next series of questions.
Please.
So Lauren Smith wants to know about climate change, and will that drive an explosion of evolutionary changes in fishes?
Yes, I think it will. And that's hard to predict, but let me answer the question this way. We are catching fish in Long Island Sound that we would only see occasionally, perhaps after a hurricane. So some species are becoming a little more common. As the water is warming, they're moving up north. A few years ago, the power plant near Clinton, I believe it might be, in Connecticut, the nuclear power plant, basically had to build a longer straw to bring its coolant into the plant. I'm just saying this if there's anyone thinking, oh, it's not getting warmer. It's getting warmer. And I'm telling you, there's no other bunch of nerdy, precise, super safe engineers on the planet than there were people who were designing nuclear power plants in the 1970s. And they had to project it out, like what kind of straw do we need to bring coolant in at what depth? And all the scenarios they had at hand, they built it the length they did to the depth they did. The change was unanticipated, and it happened. So they had to build a bigger one, a deeper one, so that they could cool their reactors. So fishes and aquatic life are changing their distributions. Now, the problem, think about it. If you're a tropical species, awesome. The warm water band increases. But if you're a pole adapted species, bummer, because that band is shrinking. And those Antarctic fishes I was talking about, if you take them to 0.8 degrees centigrade, so 0.8 over 0, they die. So they can't handle the heat stress. And they have nowhere to go. It's not as if they can move further south to get to cooler water. So we're going to see changes in distributions. King crabs are invading the Southern Ocean, coming from Southern South America. And basically it's a smorgasbord buffet, because invertebrates in Antarctica have never really had crushing predators. So most of them are soft-shelled and soft-bodied. And these king crabs are like, yeah. I mean, they're just, decimating communities.
Well, and it reminds me of the news that we heard recently coming from the Pacific Northwest of all the, not necessarily fish,
Yes.
but it was something like a loss of 1 billion sea creatures,
marine organisms, right?
Yeah.
So to the question though, where it's really intuitive, is that going back to the Doonesbury cartoon, the reason why the multiple resistant strain evolved is because we've been zapping tuberculosis with antibiotics since Fleming found a fungus he didn't want to grow on a petri dish. And that sense, this will be a large sieve in my mind's eye that will act on the present genetic variation, that will result in some evolutionary change that, yeah, could be setting the firmament up for evolutionary change that would be really noticeable on more deep time situations.
Okay. We have some people asking, I think, questions that are really tied to their sporting interest in fish, and also our passion for certain kinds of fish that
Please.
we enjoy eating. So any suggestions on how to revive the striped bass population? Is climate change affecting it as
Yeah,
well?
it certainly is. And it's a problem of overfishing, but I'm confident that management practices will help that. Fisheries biologists are really talented scientists. They really know their organisms, and both their hearts and their minds are in the right place on these issues. They are the best conservation biologists on the planet, in my humble opinion.
Okay. I want to move to, there are a bunch of questions on the antifreeze containing fish. So I'm trying to sort of digest all of them.
So Lauren, can I just jump in?
Yeah.
So I see Stephen Broker is asking, he has a textbook from 97 that has 24,600 species. There are about 500 species of fish described every year. New species.
New species.
So that's why we've gone, basically added 10,000 or so species in that time period.
And that's fascinating because, you know, according to what we've learned from your presentation, there are probably many species disappearing that haven't been described yet.
That's true. And that I call it's the 1-2 punch of the biodiversity crisis. One is we're losing biodiversity, and we're losing biodiversity that we don't know about.
Exactly. Okay, I do want to go to some of these antifreeze questions. So Josie wanted to know, going to the glycoproteins, do these create a more stable cellular environment, or does the cold cellular environment affect protein folding kinetics?
Yes, so that's a great question. And it's thought that the proteins are adapted for the kinetics under the cold conditions. It's not been demonstrated the antifreeze beyond freeze avoidance is involved with that.
Okay, and Michael wants to know what role krill play in the
Wow.
story of antifreeze containing fish, or in the current Antarctic Ocean.
Krill are the pasta of the Southern Ocean. They are super abundant, dramatically over harvested, and they are the keystone resource of the Southern Ocean. So ultimately it's tied to photosynthesis, because the krill are eating algae during the months where there is sunlight. And the krill are consumed by a wide range of organisms, different body sizes, from whales to these small fishes. That is, the krill goes the entire ecosystem.
Wow. So, so Rhoda has asked which North American populations of fishes are in the greatest danger, evolution, ecosystem changes, etc. And I think kind of what you just said connects with that question, in the sense that there are certain populations we need to make sure that they're not in danger because everything is reliant on them.
Correct. It's a little different with the North American fish, though. So the reason why each of these entities, geopolitical entities on their own, have the diversity they do. Okay, so I'm going to Tennessee, Alabama, Kentucky, Missouri, and Arkansas. Each of those states have more species of freshwater fish than all of Europe on their own. So the reason, in part, is not just there's a lot of species, but they have a lot of species that have very, very tiny geographic areas. And this is not the result of 21st century, oh, here's a new species, I'm going to keep splitting it. Most of these were discovered and described in the 19th century and first half of the 20th century. So the idea is that if you have a small river basin, that has a lot of species, but also a lot of species that you find nowhere else in the world. It's called being endemic. And you nuke that river basin through, say, cattle farming or agriculture runoff or urban, impacts, then you're removing a lot of evolutionary diversity in one fell swoop. So that's the danger there.
All right, so two things. One, the population of fish that everything else depends on. And two, the possibility of really doing a lot of damage to a broad population of fish in one fell swoop.
Correct.
Yeah, I think it's really hard for most people to wrap their minds around how sort of interdependent our environment is and how connected fishes are to it. And that's what I love about this topic.
And how unique space is. So I'm not a conservation biologist, but my work is used a lot for the conservation of Eastern North American freshwater fishes. And the language people will use for landowners is that the fishes in the stream on your property are found nowhere else on the planet. Nowhere else. You can't even go 2 counties away and find that species. And I got to tell you, more than half the time, people are like, wow, I'm really proud of that.
I should also rethink my use of Roundup on my lawn.
Exactly. Or allowing the cattle to actually go into the stream, or, 20 other things, or just allowing a riparian area to grow. Don't farm all the way up to the edge of the stream. And, there's a lot of great NGOs as well as some government entities that allow that facilitation and allow some remuneration to these landowners too, which I think is important. I think property rights are an issue and shouldn't be blown off or dismissed. And the fact that, many of these stakeholders are multi-generational entities, live there for a long, long time. So that book I recommended, The Snail Darter and the Dam, talks about just that, where the Little Tennessee River Valley was one of the first areas settled by Europeans west of the Appalachians. And a government agency came in and took all their land away to build a fake lake. And not only was that an impact on the biological diversity, but it was an impact on people's rights and the cultural diversity of the area. So it's all connected, I think.
Absolutely. Okay, I want to go to more questions coming in as we talk.
Yeah.
This is a great conversation. For the divers in our audience here, David wants to know, we're told that fish cannot see color, but when we dive, we see brilliantly colored and patterned fish and color vanishing as we go deeper. Is there an explanation?
Wow, that's great. That's a great question.
David Saybrook, class of 1950, by the way.
Oh, wow. Say what, right? Okay, so fish do see color. And we know this because how we perceive light, there are proteins in our retina called opsins. And we have different opsins. And the different opsins are attuned to different frequencies of light. So if we want to know what a fish can see, we can, we could take a retinal tissue and zap it with light and look at the reflectance spectra of the light coming off. So most fish can see color. In fact, most fish can see the UV. Now, the phenomena you describe is, wow, spot on. And so color attenuates with depth. So where you tend to find the brightest colored fishes are in habitats where predators are not going to mess with them. So on coral reefs, if you're a brightly colored butterfly fish, and if you've noticed the behavior of colorful laterally compressed reef fishes, they're really good either at just fast burst swimming, or they could be crepuscular and they could go into complicated structure to avoid predators. Or most colored fishes are in water depths that are too shallow for predators to be. So here's the conundrum: is what is driving the color is you're not going to find colorful fishes that are deep, because why would you expend all that metabolic energy making these colors, whether it's structural colors like bird colors, it's the structure of the scales, or lay down pigments, melanophores and chromophores they're called. when your colors are not going to work. So exactly what you observed is what we think is driving the distribution of colored fishes. So the deeper you go, the colors aren't even working. And if the colors are there to attract individuals for mating or for intrasexual conflict, like males fighting over nesting sites, whatever the behavioral signals those colors are serving, the colors are not going to work at deeper depths. So you tend not to see them there. So there's this trade-off. If you're going to be colorful, you got to be in areas to avoid predation, and you got to be in areas where the colors are going to work, where the signal will be received by the intended recipient.
Okay, so just talking about the fact that fish can see color, I'm really curious about this news story that was reported a few years back about the self-awareness test for fish. Are you familiar with, or can you speak on that? The idea that the fish could identify itself in a mirror.
Yeah, I mean, tangentially familiar with that, and I'm not surprised. So there are groups of fish whose cerebellum has the same ratio to a body mass as humans do, and feel free for someone to insert a joke here, but say you know that person, but it's a group called elephant fishes. They're freshwater, found in Africa, and they look really bizarre. They have a long snout and they have a big head, and they communicate with electroreception. So they have, many fishes can generate electricity. They generate basically low voltage, low amperage electricity. And they do that for individual recognition. So you have many different species of elephant fishes that live in the same muddy water habitats in freshwaters of Africa, and hybrids are very rare. So they're finding each other. But this is a long-winded way of saying, I'm not surprised by the self-recognition, because these mormyrids, by the way, there are people who study play, P-L-A-Y. I'm not one of them, but there are people who study that. And there are criteria of what constitutes play. And my understanding is elephant fishes play. And so where my thinking is, if a fish can play, certainly a fish would have the central nervous system capacity for self-recognition as well.
Okay, and that goes along with Christine's question about cuttlefish, that they've been reported to have exceptional intelligence. And Christine wants to know, are they related at all to true fish?
So cuttlefish are mollusks. So they're related to squids and clams and mussels and snails. But mollusks are really interesting in this regard. They have highly, I think, advanced ability, sensory ability, and conceptual ability, I guess, for lack of a better word, thinking on my feet here. So next week, my colleague, Professor Casey Dunn, will be hosting a screening of the Octopus Friend film. And it's clear that there's a cognitive ability in cuttlefish and squids and octopi that's really impressive. Now, I'll just add a biological nerdy fact to this, that what I find so fascinating is in vertebrates were able to invent really narrow nerves. And the vertebrates were able to invent that by laying down a proteinous sheath on the nerve fibers called myelin. So it's like a wire that has an insulation. The mollusks never figured that out. So their nerves are giant. And in fact, the Nobel Prize winning work in the late 30s, early 40s on the action potential of nerves, like basic physiology of nerves, was done on squid because the nerves are so easy to manipulate. They're so big. So here you have this different engineering of the nervous system. And it's not like a brain encased in a bony head either. That's something that kind of defines vertebrates, that yet this cognition has evolved independently in these two different lineages, mollusks and in vertebrates.
So talking about the sort of nerve structure of these two different lineages, Robert wants to know if fish feel pain.
Yes, fish feel pain. Absolutely. And that is just, I mean, no one's ever asked a fish, but if people look at hormonal responses to pain, there's a signature, and it's, put it this way, if you were to take an automobile, any automobile, and you see like, yeah, it's a V8 internal combustion engine, you know it has a carburetor, right? You have to have a mechanism to vaporize the fuel. And if you look at a fish that has all the parts that we have and all the aspects of sensory biology we have, so just as a premise, you would infer that they feel pain.
Okay. Well, we're coming up to the end of our presentation. Still a lot of questions unanswered. I think I'm going to close with one that I think rounds us out to your opening premise. You've really convinced us that people are just sort of large walking fish. And so I'll close with Linda's question. What fraction of our DNA is shared with fish?
That's a great question. Okay, so it depends on what fish we're talking about. So most of the fish we talked about today are called ray fin fishes. That's the big group of fish. Then the other fishes are lungfishes, lamprey hagfishes. They don't have jaws, so we don't need to talk about them. And sharks, skates, and rays. Most ray fin fishes are another group we call teleost. So of the 35,000 species of ray fin fishes, only 50 are not teleost. Teleost duplicated their genome prior to their diversification. So most teleost have multiple copies of a given gene. The non-teleost ray fin fishes only have one copy, like we do. And so for those non-teleost ray fin fishes, the matchup gene to gene, that's called synteny, is really high, really high. It's amazingly high. So the genomes, in terms of the genes that compose the genome, are highly conserved. Now, if we're looking at, if you go to a single gene, how different is the DNA? We've all heard, oh, you know, our DNA is only 2% different from a chimpanzee. They're talking about go DNA like site to site, how many of those sites differ. In terms of genes, we have all the genes a chimpanzee has and vice versa. And we almost have all the genes that these non-teleost ray fin fish have and vice versa. But in terms of how different the individual DNA are, it's pretty high. And it reflects the fact that our time to common ancestry to those fish is about 380 to 400 million years ago.
Fascinating. I think we could probably talk for another hour with all of these
I certainly
questions.
could, but I don't, but thank you so much. Yeah.
Yeah, this has been really fun. And for those of you who are looking for more, thank you, Professor Neer, for mentioning that on Monday we'll be doing the screening of the film My Octopus Teacher, and a fellow curator from the Peabody, Casey Dunn, will be answering our questions. So we'll get to talk about invertebrates. And I think Michael is saying something here that we would all agree. He's saying, I'd love to take your course. So we do hope that we'll be able to offer more programs with you through Yale Alumni.
Thank you. I would love to do that.
Yeah, and I'll just give you closing word, anything you would like to say as we finish today.
Yeah, just thank you all for your attention today. And I'm, my email is thomas.neer@yale.edu. I'm easy to find. New Haven's not only a small town, but Yale is a village. And I'm on Twitter @TJNeer, and I follow back. And if you have any questions, please don't hesitate to reach out. I love interacting with our Yale community.
Awesome. Wonderful. Well, have a great day, everyone. Thank you for tuning in, and we will see you at the next Yale Alumni Academy Festival of Ideas and Enrichment event on Monday. Bye-bye.
Bye, everyone.